Oops, I believe we are live, aren't we now?
Yes, hi everyone. We have just gone live. It is my pleasure to welcome you to this live Q&A session, the very first one for today. We've got Ivan Lukic from Partech, Marcus Tyndall, University of Reading, and Niels Tomerup from the University of Copenhagen joining us. We've received quite a few questions actually for this Q&A, so I'm very excited to hear your answers. The first question is for you, Marcus. What is the effect of not including spatial effects in mathematical models? Marcus, you are muted. If you can just unmute yourself. Quite a few questions actually.
Sorry, there's a recording playing back for what you just said a moment ago.
The first question is for you, Marcus. What is the effect of not including spatial effects in mathematical models?
Hello, can you hear me all right?
Would you be able to just have the Zoom meeting
Marcus,
open?
you are muted. If you can just unmute yourself.
Quite a few questions actually.
Sorry, there's a recording playing back for what you just said a moment ago. There's a major delay here.
What is the effect?
I don't know what's happening, sorry.
Not including mathematical models.
Hello, can you hear me all right?
Marcus, can you please close the Swepcard platform and just have the Zoom platform open?
Okay, right. Okay, that might fix that.
Yeah,
okay, right. Sorry. Yeah. Right. Sorry, apologies. Right. Yes, sorry.
Yeah, that's no problem. So on to the first question. Would you like me to repeat it?
Yes, that would be good. I was just trying to work out what was going on then.
What is the effect of not including spatial effects in mathematical models?
So it depends very much upon, thank you for that question, by the way, and to the person who raised it. It depends very much upon what the question is you want to answer. So if you do have spatial resolution, you wish to delineate between different areas of the cell, in doing so, then you need to incorporate that into your model realistically and appropriately. If you don't do so or take account of it in an appropriate way, then you may find that all of a sudden you're not accounting for spatial variation where, for example, two proteins may not actually be in the same place at the same time. This is where we talk about spatiotemporal effects. So it's quite important then, if you realize this, that you take into account the spatial difference in that way. Now, that's where partial differential equations, you can also do other things I didn't mention in my talk, or agent-based type models to do things in that way. So it's quite important.
Brilliant, thank you very much. Ivan, on to you now. Can you analyze multiple tissue slices samples together, or is it one sample at a time?
Thank you. It's multiple tissue slices that can be analyzed at the same time within the same project in Partech Flow. That has been shown during the presentation, and I believe it's also available on, you can watch it on demand. So if that person, if the person who asked the question would like to see it again, they're more than welcome to. Thanks.
Great. Thank you very much. Marcus, the next question is again for you. How much data is needed to inform a model?
The more the better, but actually with the sort of mechanistic modeling you have, you can actually approximate a lot of the, for example, rate constants and diffusion constants to begin with. So there is data out there for other cell systems if it's not available for your cell system to get you started. If you then identify by doing the modeling that, for example, diffusion coefficients or other parameters describing processes really need to be described in more detail, then that can help identify where experiments are needed. That's also one of the strengths of modeling, is the fact that you then can test the model beforehand as to what are some of the major mechanisms that are affecting the outcome you're interested in. So you can help direct your experimentation and also reinforce your modeling in that way.
Great, thank you very much. Niels, we've got a question for you here. TADs may differ from cell of tissue types. How does this influence the functional interpretation of a given breakpoint?
Yeah, that's true. So we used for our global analysis of distribution of breakpoints, we used TADs from fibroblast, IMR90 fibroblast, but even if you select from another tissue, you will have, they will either be the same or they will be overlapping. So you will identify the same loci, but what may differ, of course, is the extent of how far out the TAT boundaries may be situated. But the specific target gene which we believe drive this high risk for removal of regulatory elements, they will of course be situated in overlapping TAPs, irrespective of which tissue you look at. And I think one of the ways to solve how far out a specific regulatory landscape goes is actually to look at evolution, because these regions have been, or rather the enrichment of these non-coding elements in any type, I mean, it could be VISTA enhancers, it could be sequence-based conserved elements or whatever, they will actually probably tell us exactly how far out can we expect to see a phenotype of so.
Great, thank you very much, Niels. Ivan, and now over to you. It's multiple questions. Please let me know if you would like to answer them one by one. I think they are all connected to each other.
Please go ahead.
How much can you look under the hood? Your own scripts are written in what language? Do each intractable object come with explanation and help? When you are doing an analysis, can you see the code and fine-tune it, change some parameters?
Okay, so this is a bundle of questions, but you were right. It does make sense to tackle them at the same time. I haven't been, so we've been limited with time, obviously, for this presentation, and I was able only to scratch the surface. However, if you would like to know more, I invite you to take a look at our webpage. So it's partech.com, obviously, and you will find the recordings of our past webinars. And now some of those actually show what's going on under the hood. For the current session, I've been showing only this visual part, data viewer, but you can also see what a pipeline looks like. It's not a black box, and you can first access each data from each individual step. For instance, when you quantify and you generate gene count matrix, you can download that matrix. If you filter the matrix, you want to remove, for instance, mitochondrial genes, or you want to remove some samples, you can again export the result. Now, this is for the results. About the processing tasks. For each task, you get a summary of the tasks, so key details of what has been going on. For instance, if it's about normalization, how exactly were the data normalized, what was the transformation that has been used, and you can also ask to see what exactly was you can ask for the output of the command line. So what has been executed on the command line. We do have interactive help. So there are, for instance, tooltips to guide you and explain any quick, so short explanation of any tool. All the tools can be fine-tuned. For example, if it's it doesn't matter if it's one of our own tools or an open source tool, all of them can be fine-tuned, so you're not limited to, for instance, only some default settings, but you can customize to your own needs and your biological question. And there was, I think, an additional question about the package itself. We use several different languages where actually different parts of the package use. If you have any additional questions, here I'm still here. Yeah.
Great, thank you very much. Niels, this question is, we received this question for you. How can you be sure which gene that is the dysregulated target by a breakpoint within a given TAD?
Yeah, that's a really good question, because because obviously there could be, even if these high-risk TATs are gene poor, they do contain or harbor other genes, including protein-coding genes, and well, what we see this striking enrichment in the known long-range position effect loss of homobox genes, transcription factors, nuclear receptors, and other key developmental genes. So that might guide you towards a candidate within a specific TAT. Experimentally, of course, you would expect, if you remove key regulatory elements, that you may alter the expression pattern of the target gene. For example, you will see reduced expression. But of course, there's a problem there, because other genes in the same TAT maybe also be, they maybe show either co-expression or also may also be dysregulated. And of course, another probably most important problem is that many of these genes are very tissue specific. For example, we know from the phenotypes that most of them affect the brain with intellectual disability or autism spectrum disorders. And so the genes may not be expressed in the tissue you have access to. And of course, one way to solve that, but that's of course not trivial, would be to make iPS cells and organoids and so on, and look at, for example, brain progenitor cells from the patient itself. But that of course would apply considerable experimental studies. So it's not a trivial thing, actually.
Great, thank you very much. The next question is actually for all three of you. Marcus, I know that you are in a bit of a rush, so if you would like to go first and answer this question. The question is about spatial biology in general. What excites you about spatial biology? Why is it such a great area to look at? And how do you think the future looks like for this area?
I think it's quite exciting. I think with the developing data and the new experimental methods that have clearly been appearing in the last 5 years, there's a lot of opportunity here for developing more spatially realistic models and accounting for spatial temporal behaviors. So I'm quite excited. I think also it very much depends upon the questions people are wanting to answer. So I think it's important to take sometimes off when I talk with people in industry and also academics doing modeling, it's important to take a step back and think, okay, what am I planning to do here with the model and my data and what I want to be answering scientifically at the same time? And those three areas we can really Venn diagram overlap to make sure there is consistent use of the modeling, because sometimes people get very excited about modeling and say, okay, we need to do this and use modeling. And actually you turn around and say, well, actually experimentally you can show this okay, so it's not relevant for a model. So it's very important you get those three connections working well. I do apologize. I have something unexpected that just popped up today, so I'm going to have to disappear, but thank you to everybody for their time.
Thank you very much, Marcus. Thanks very much for joining us.
Bye-bye.
Bye. Ivan, Niels, what are your views on this, on spatial biology and its future?
Well, I think one of the sort of academic questions that I found fascinating is how many unknown type of cells do we actually have in the body? Because we are already, new very specific subpopulations of cells are emerging from via different techniques, expression pattern or whatever. So probably we will learn a lot of new things, which it's like astronomy. You see new galaxies and stars and whatever. So I find that quite fascinating.
Great, thank you very much. Ivan, it's your turn now.
Yes, thanks. That's an interesting question, and I like Neil's analogy of astronomy, or maybe in general physics. So if you talk about the general model, then when do we stop? Is it an atom? Is it a basic particle, a nuclear particle, or do we go further? So it's also with spatial. So we started a long time ago with bulk tissue analysis, and then there was single cell analysis, and now we are going one step further with the single cell within the tissue. And then and we've seen over the past 3 days, we can go deeper than single cell. So it will not only be the events on the single cell level, but on subcellular level within tissue. So certainly the future looks bright and very exciting, but I would also like to go back to the Marcus's point. It really goes, yeah, it's fashionable and it's great and it's exciting, but one really does have to go back to the biological question, like, do we need it, or can we answer it in using a simpler approach, or do we need this, and how far do we go?
Mm-hmm. Great, thank you very much. Niels, we've got one more question for you. Are there any of these constitutional balance rearrangements recurrent, for example, like in cancer-associated translocations?
Well, with actually 2 exceptions, no, they are all unique. So each of these is a rare event. It's for the translocations, they are unique, and what that means, since we see them in 1 in 500 in the population, this means that there are tens of thousands of them out in the whole population anywhere in the world, and only some 80% of them will be not be associated with disease, as far as we know. But our, you could say, limited mapping, where we have just mapped now about 600 cases, controls and cases together, really suggests that these are the closest we get to random mutations in our species. So, and since they have this unique thing that you can truncate genes, and genes truncation events, of course, is a rather high risk event, in the sense that it doesn't matter whether you truncate the coding part or the intronic part. Still, genes occupied about 40% of the genome, and we see exactly that these rearrangements truncate a gene in about 40%. Both in controls and in cases, and it's just a matter of which genes. And then they have this added bonus that they also will have effects by rearranging the topological domains. For inversions, we actually see some founder inversions, because they have they have a higher genetic fitness than translocations, and so you actually see them in larger families. And we also see for example, in Denmark, we have, by screening mapping all available inversions in the country, we have found 13 founder mutations, where we see the same mutation in different small families that we cannot link by pedigree analysis, but where we actually can link them by genomics. And since it's a kind of random mutation screen, then I have also advocated for many years that all of these cases, whether they have a phenotype or not, should be mapped. It's so easy, it's so less costly, and it will tell us a lot about the effects, not just in relation to specific truncation events, but also in relation to the regular setup of the human genome. And this is done, it's actually all cytogenetic lab will encounter these cases now and then, for example, in intellectual disability, but also in infertility. And it's just a matter of trying to, you could say, do it in a systematic fashion. But of course, it needs funding.
Great, thank you very much. This brings the Q&A session today to the end. I just wanted to thank you very much, Ivan and Niels, once again for joining this exciting session and also for these wonderful presentations that you delivered. Ivan is here with us for the rest of the day. Please feel free to reach out to him and also Pátek if you would like to discuss this area further, as well as Niels. I'm sure they will be happy to chat with you. Thank you very much and enjoy the rest of your day.
Thank you.